I will explain few examples, right. I will show us few examples now. If you have a sequence, right, for example, this is a sequence ATA, TAC, GGG, CAG, GAGC, right. So, this is example. So, what is the AT content or what is the GC content, right, because this is very important, because some if you take some genomes, they are highly rich in AT content and some cases highly rich in GC content. And if you look into these genomes, if there are highly AT content, so they are highly flexible, and there is some cases GC content, they seems to be little bit rigid. So, and also there are places where we have more AT sequences, right. These sequences, they have high possibility to interact with the protein side, because they are highly flexible. So, they can change the conformation and they can have high probability to interact with the proteins. So, this will give you an option how, where are the flexible sequences or the rigid sequences, and the AT content or GC content will give you the information. So, now this is the sequence. So, what is the GC content? So, for this case GC content means what is the percentage of G and C in the particular sequence. For this, first we get the number of G, right. So, how many G's here? 1, 2, 3, 4, 5, 5 G's, the number of G equal to 5. Then number of C's, how many number of C's? 1, 2, 3, 4, right, number of C equal to 4. Now, what is the total number? 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, right, 16. So, GC content you can calculate, out of 16 how many G and C, this is equal to 5 plus 4, 9. So, 9 by 16 into 100, this is equal to 56.25%. How to get the AT content? How many number of A's? 5, number of T's? 2, okay. So, AT content equal to 7 by 16, this is equal to 43.75%. So, you can do that, because easily you can subtract 100 from this one, you will get this, right. Okay. Now, also there are, we can derive various sequence based parameters. Okay. So, if you see the nucleotides, so they have various features, right. You can calculate various features based on this sequence. So, I explain few aspects, right. So, one is DNA rigidity, right, or DNA flexibility. Right. So, totally how many bases? If DNA 4, right. So, if you take the dinucleotides, there will be 16 dinucleotides, but if you consider the pairing, then 16 you can divide, you can reduce to 10, right, 16 reduce to 10. For example, if you see this AA, A is this one, if it is CA, CA is similar to right, if it is AC, right. What is the complementarity? TG. If you take a reverse, GT, right. So, if you see the AC and the GT, right, both are same, right. Likewise, if you take the another one, for example, if you take the AC, or you take the CA, right. CA means this is equal to GT, so TG, right, this is 9.34 and TG is 9.34, right. If you see the how many unique pairs? 12. So, AG and CT, right, this will be same, and this is 7.05, this is AT, right. You are getting the same, so you will get the same one, right, if you the TA and AT, right. If you take AT, what is the complementarity? TA. If you reverse AT, you get the same, right. So, you will get about 10 pairs you will get, right. So, this is for the di. If you go the tri, totally how many possibilities? 64 possibilities, and due to this complementarity you can reduce this to 32. This way, if this is the dinucleotides, you can have these pairs, right, but here the trinucleotides, you have the out of 64 you have the 32 cases, right, here. Okay. So, now first we got the flexibility, how you can either use the dinucleotides or the trinucleotides. What is the advantages of using dinucleotides and trinucleotides? Dinucleotides have less information, because we have the less number of possibilities. Trinucleotides have more information, because AA with A is we can get more information, right. So, the trinucleotides can provide better information than dinucleotides, right. So, we provide trinucleotides. Then what will happen with tetranucleotides? More information, but what is the effect? What is the problem? What is the issue with the tetranucleotide? Like amino acids, right. If you got how many amino acids? 20. If you go with the pair, 400, right, 20 to 20. If you go with the tri, 8,000, but data set is same. If you want to derive the features, we use the same data set, for example 1000 or 10,000 sequences. If you use the di, you will single amino acid case you will get more number of data. If you go with the di, then the data distributed. If you go with the tri, then it is very sparse, right. Some case you will get zeros for many cases, right. Likewise, the nucleic acids, if you take the di, we get good number of data, but tri provides more information, and go with the tetra, we will get better information, but the number of data are less. So, the values you obtain for each tetranucleotide is statistically not significant, because here even trinucleotides, if you see some cases you get very less number, right. See some cases we see 1, 2 like this. So, if we go with tetra, then the data will be less, right. Okay. How to get this one? For the case of DNA flexibility, there are various ways to get the flexibility. Okay, you can consider the DNA as a rod, right. So, put the one base, second base and third base, right. Then you can bend, you can apply a force and you can bend it, right. Then you can see the same force you apply for all the trinucleotides and see how far they can bend. Based on that, you can compute the flexibility or the rigidity of each trinucleotide. If you have the 3D structures, where shall we get the 3D structures of the DNA? In DB or the PDB, right, nucleic acid database or the protein data bank, right. So, if you take all the trinucleotide steps, right, take the whole DNA structures and cut into pieces, right, even you have the overlapping pieces, right, all the trinucleotides you take, right, and then if it is completely straight, right, you can put one stack one on another. So, you know the width of the diameter of this DNA, you know the thickness, you know. So, you can see this is 7.2 angstrom, but if you see the structures, that is not straight, it is bent. So, in this case, based on this bending angle, you can this related with the flexibility, right, because if you know Young's modulus, what is Young's modulus? Stress by strain, right, E equal to stress by strain, right. What is stress? Force per unit area, because we know this is circular rod cylinder, that you know the radius. So, in this case we know that we can calculate the area. Force also we know how much force we give, that is 480 piconewtons, right. If you make it constant, because we use a constant, so in this case it does not matter, right. So, it is estimated to be 480 piconewtons, right. So, now we can calculate this. What is strain? Yeah, increase in dimension divided by original dimension, right. So, if it is without any change, then this is straight. So, now in the 3D structure you can calculate the distance from the first one and the fourth one. So, you can calculate the distance, right, with the coordinates x and y1 z1 and x2 y2 z2. So, then calculate the distance. Now, from this distance and the original distance you can calculate the difference. So, from this difference you can calculate the rigidity, right. So, then this is the direct way to get the Young's modulus. So, based on that, for all the different types of trinucleotides you can calculate the Young's modulus. This is the inverse of flexibility, this will give you the rigidity. So, this is how we get this data. So, we take the set of nucleotide structures available in the data bank and cut into trinucleotides and get the dimension, how they change in the distance, and we convert these values to calculate the strain and stress we know. So, stress by strain is the Young's modulus. So, we know the values, the Young's modulus, that is Newton per meter square, these are the values. So, now if you see these values, can you tell few nucleotides which are rigid and some of them are flexible? For example, ATA, okay, it is ATA, right. ATA, what is the value for ATA? ATA, this is 2.36, this is known to be flexible. Also, if you take the CCC or GGG, that is rigid, right, CCC or GGG here, okay, which is the value, okay, this is 6.07, this is rigid. So, if you compare with the known values, the expected values we know, most of them match, and the main problem from this table is some cases we have less number of data. So, now if you have more number of nucleotide structures available, so you can use all these structures, so you can refine this. Some cases it is okay, it is 27 or 49, but some cases it is very less, say 1 or 2. In this case the data are not statistically significant, you have to go for more number of data, right. But anyway, you can use this as a source for the stiffness of each trinucleotide. This is the one example for the hydrogen bonds, the calculation of hydrogen bonds. This is for the flexibility or rigidity, and also we have data for the base taking energies. What is the base taking energy? Yeah, if we have the two bases stacked to each other, right, what is the energy, right, produced between these two bases, right, two things. So, you can calculate this using any potential, Ampere potential or any potentials. You can use the different types of interactions like van der Waals interaction or electrostatic interactions, right. And if you put these two bases and then you can calculate the energy between them, and you can estimate the base stacking energy. So, here I show the data for the different dinucleotides. Okay, here this I give the data for both DNA and RNA. So, you can see ACGUT, here also ACGUT, okay, because U and T are not coming together, this is why I put dash. So, for each cases A and A or A and C, okay, so A and G, A and U is 1.33, A and T is 1.83. So, you have the table for the base stacking energies, okay, fine. So, now we have different these tables, also we have a set of values for different parameters, okay, that maybe next time, next class when we discuss about database, I will show a database which contains the information regarding different values, right, say hydrogen bond energy and the melting temperature and so on, right, fine. So, now if you have a sequence, how to calculate the average property of any DNA sequence, that is the question. Okay, if we take any sequence, I will explain with two examples, one is with the rigidity and one is with the base taking energy, right. So, if we take this sequence ATA, TAC, GGGCA, right, here we need to divide this into overlapping segments. Here data are known for the trinucleotides, right. So, we can classify into overlapping segments, right. So, what are the different overlapping segments? If we take the trinucleotides ATA, right, ATA, and from here TAT, and from here ATA, and here TAC, and so on, right. So, totally how many nucleotides here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 nucleotides, right, 11 nucleotides. So, how many possibilities you will have? 9, right. So, 9 trinucleotide steps, right, and then take the values from this table. So, what is the value for ATA? ATA is 2.36, right, 2.36. Then what is the value for TAT? The same 2.36, ATA same 2.36, and TAC 2.97, right. So, add up all the values, then finally you will get 31.74 and divide with the total number of possibilities, that is 9. So, 31.74 divided by 9, this is equal to 3.53 in 10 power Newton by meter square. So, you will get the exact average rigidity, right, for this particular trinucleotides, particular DNA sequence. Okay, now if you want to have the base taking energy. So, base taking energy we have the data for dinucleotides or trinucleotides, dinucleotides, right. Here we have data for trinucleotides, here we have data for dinucleotides. So, we need to divide this DNA sequence into overlapping dinucleotides, right. How many dinucleotide steps you will have? N minus 1. So, this is equal to 11. So, total you will get 10. So, now how to classify this AT, right, AT, then TA, then AT, TA, AC, and so on. So, what is the value for AT? 1.83 here, 1.83, TA 1.83, AT 1.83, TA 1.83, and AC 1.73, right. So, add up all the values, that this will come with 16.31 divided by this n minus 1. So, there will be 10 dinucleotides. So, divided by 10, this will give 1.631 kilocalorie per mole. Right. So, now if you have set of sequences, you can calculate the values and you can compare the sequences, which sequences are rigid, which sequences are flexible, right, and what about different types of energies. And from this you can calculate, right, various structure based factors, or whether this region is able to interact with the DNA, this particular protein and so on, right. You can see the DNA interactions or the DNA drug interactions and DNA protein interactions and so on. So, this you can calculate, right. Then how to derive these parameters using computational tool, right. Then we can develop a program to calculate these steps, right. How to do that? For example, if you have a sequence, can you tell the sequence A, T, G, G, C, A. Okay. So, how to calculate the base stacking energy? So, the first question is how to get the base stacking energy. What to do first? So, first we get the sequence, right. We need a sequence. So, now the overlapping dinucleotides, right. So, we have the overlapping dinucleotides, right, and then so we need the table, right. We need the base stacking energy values. Energy values for all dinucleotides, right. And then what to do? Yeah, so we have to assign the values, right. So, assign the values for each from this step 2, right. So, assign values, right, in the step 2 to this step, right, and then take the sum, take a sum and then divide by total number of dinucleotides. So, what to call how many number of dinucleotides? N minus 1, right. So, here this will give you sigma I, the equal base stacking energy, right. So, you can put GBS, right, of I. So, I is the number of dinucleotides, right. So, I stands for the dinucleotides. So, add up, then here divide with this N minus 1, right. So, N is the total number of nucleotides. So, we will get the values. Likewise, if we have the flexibility, what to do? Similar way. How to get the flexibility? The same way, right, or rigidity. So, this is what to do? Only the same procedure you have to follow, only the difference is instead of die you have to move to try, right. So, get the sequence and make the sequence into overlapping trinucleotide sequence. So, you have to give the overlapping trinucleotides, right, and then have the table and assign the values and add the values, right, sigma E I, right, I equal to 1 to n, where n is the number of trinucleotides, and they divide with the n minus 2, because trinucleotides, so n minus 2. So, you can do the same way and you can calculate rigidity. So, depending upon the data, whether you have the data for the each nucleotide or the dinucleotides or the trinucleotides, right, you can assign the values and you divide, you have the overlapping trinucleotides, right, then you can calculate the rigidity. So, why we need to derive these parameters? What is the importance of this? If you do see the rigidity values, the rigidity values have some information to do with the binding affinity. So, if we have the proteins, right, and interact with the DNA, so right, if you have the proteins and the DNA, so you know the binding affinity. So, there are various databases available which will give you the binding affinity of protein DNA complexes. Sometimes you have the same proteins interact with different DNAs. In this case, the affinity is different. So, take the DNA sequence, because protein is same. So, difference mainly comes from the DNA sequence, right. So, in this case you have to look for the contributions from the DNA. So, you get the DNA sequence and then see the various parameters, for example, rigidity or flexibility, right, and how this flexibility or rigidity, right, varies with respect to the DNA sequence. And we have the delta G values, right, that is free energy of binding, and compare this delta G with the known sequences, known E values. So, for example, here delta G 1, delta G 2, delta G 3 for different complexes, right, protein DNA complexes. So, this is Y. So, you have the X different parameters, for example, rigidity, right. So, you have the rigidity or any types of energies, right. You can compute from the sequence, this you can calculate from sequence. This is the delta G, we know experimental, this is experimental. So, we have the X values and we have the Y values, we have set of data. So, you can plot, right, this is the delta G, this is the any parameter, right. So, you can use any parameter or any property, right, say rigidity or the flexibility or the binding energy or hydrogen bonding energy. So, then you can plot. When you make a plot connecting each parameter in delta G, and you can see that which parameter fits very well with the binding free energy. So, this will be helpful to understand what are the various factors which are important to understand the binding affinity of the protein DNA complexes, right, you can see that. So, on the other hand, you can do with the protein side also, depending upon the different types of complexes. If the complex varies only in the DNA side and mainly the DNA part is important. If there is different protein complexes with different proteins and different DNA, then you can use the parameters derived from the protein side as well as DNA side. DNA side there are various parameters, right, maybe in the next class I will discuss about database which contains more than 50 parameters, right. So, you can use different parameters and see which parameter relates well with the binding free energy, or with a combination of parameters. We can combine different parameters which can be used to understand the binding affinity of protein DNA complexes. If you see some correlation, right, between the rigidity or the flexibility of the delta G, what can we infer?
That particular parameter is can describe the property.
Correct.
Or can predict the property, predict that property correctly.
Yeah, right. So, if you, for example, if rigidity or flexibility is important, so you can see the conformational change of this DNA, right, because of the flexibility, that makes more contribution, that is very important to understand the binding affinity. So, if you change the DNA sequence, if you mutate, then you can see how the affinity is lost, right. Then you can see how far this affinity is related with any type of diseases, right. For example, if you take the P53, right, P53 is mainly between the protein and DNA interactions, several mutations which finally essentially lead to disease, right, for example, the cancer. So, if you see change these sequences, right, you can see how far the binding affinity changes, right. So, then you relate with the different functions. Likewise, you try to see various feature parameters, right, and see which parameters are important and which combination of parameters are important to understand the binding affinity of these protein DNA complexes. As well as you can do the same for the protein RNA complexes if we have the data for the RNA sequences. Till now, what we discussed from the beginning, just we will summarize. First we discussed about the basics of DNA, right. So, DNA contains what are 3 different groups in the DNA: phosphate, sugar and the base, right. So, what is the difference between DNA and RNA? 2 prime, whether it is H or OH, and then the base, right, the uracil or thymine. So, we have 2 different types classified into purine and pyrimidine. So, purine has how many rings? 2 rings, and pyrimidine has 1 ring, right. So, this is thymine and uracil, this is the difference between the DNA and RNA. And then they form the diester, phosphodiester bonds, they have 2 ester bonds. So, this is how they have the sequence. Then why this pairing is between A and T and G and C, space constraints as well as plus the chemical groups, right. These are 2 different aspects why this pairing is important. So, if we have a single strand, how to get the complementary strand? So, you have to get the reverse inverse complement, right. You have to get the complement and then reverse it, and then you will get the reverse complementarity, right. So, there are several programs available in the literature, right. You can use the emboss is one of the suit. So, we can use it for this one, right. Now, we have the transcription and the translation. So, you get the DNA sequence, protein sequence, right, and then you have the 6 different frames, 3 forward frames and 3 different frames, right, and then we try to see derive some of the structure sequence based parameters, right. If it is available, then you can get it and then you can use it for the applications, right, to understand the functions. Fine.